Okay, morning. I would like to present this paper, Graph-regularized Dual Lasso for robust eQTL mapping. So basically, in literature of GWAS, there is one important research area, eQTL mapping, which is aiming at to find the genomic locations where the genotype significantly affects those gene expression. So typically, we have SNPs data and on a set of individuals. And we also have the microarray gene expression data for the same set of individuals. The task is to find which SNP affects which gene expression. And to fulfill this task, we can make use of the statistical test. For example, in the example shown below, each row stands for one SNP or one gene expression, and each column stands for one individual. We can first partition those individuals into groups according to their genotype of SNP. For example, for the first SNP, we can divide the individuals into two groups. And then we can do a statistical test like the t-test or NRR test to see if the mean value for the gene expression level of the two groups are significantly different or not. Then we can know if the SNP is significantly associated with the gene expression. And we can repeat this step for each SNP to see if the SNP is significantly associated with the gene expression. Alternatively, we can make use of some lasso-based feature selection method with the objective function in this slide. And the L1 penalized regularization is used to obtain a sparse weighting matrix W. So in literature, it's commonly believed that SNPs and genes are usually not interdependent with each other. And we may obtain some prior knowledge of the interplay relationships between SNPs and some interplay relationships between genes. And we can encode those prior knowledge into some networks. For example, we have genetic induction network, PPI network, gene co-expression network, and so forth. And in literature, there are already some researchers try to make use of those prior knowledge into eQTL mapping. For example, the group lasso method is used to integrate the grouping information of SNPs. And the multitask lasso are usually used to integrate the network of the genes. And the structural input and output lasso will make both of them. But however, there is some common limitations for the current methods. And the first limitation is that typically a clustering stepper is required to obtain the group information before integrate them into the eQTL mapping. And the second limitation for those methods is that they typically do not take into consideration of the incompleteness and the noise exists in the prior knowledge. For example, we all know that the PPI network may contain many false interactions and also may miss some true interactions. And the second, and the last limitation is that some other prior knowledge like the location information and the gene pathway information typically cannot be taken into consideration. In this work, to alleviate those limitations, we propose the model visualized in this slide, which make use of both relationships between SNPs and the relationships between genes. And we use matrix S and G to denote, to represent the relationships between SNPs and genes, respectively. And here, W is the matrix of the association matrix that we want to infer. Different from previous model, here we will iteratively optimize the association matrix W and do refinement on the prior knowledge S and G. And we can further integrate the location information and possible information and use those information to help us to refine the prior networks. This is the objective for the first proposed method. And we term the method as the graph-regularized Dual Lasso and use the GGD-Lasso for short. And the objective function contains three parts. The first part is the traditional lasso objective, considering the confounding factors. And here L is used to model confounding factors. And we use the nuclear norm to guarantee that L to be a low-rank matrix. And the second part of the objective is the graph regularizer, which will integrate the network information into the optimization of the associated matrix W. The last part is the fitting constraint for the prior knowledge. It's used to enforce to what extent the refined network S and G should be consistent with the initial input S0 and G0. And in order to further integrate the location information and the pathway information, we further extend the previous objective to a more general one. And here, the D is any non-negative distance measure. And the last two terms we used here is to model the inconsistency between the matrix W inferred and the prior network knowledge S and G. And the intuition behind it is that if there exists one edge in S or G, then the column and then the two SNPs are more likely to have the similar set of genes associated. And this is similar for the last term of the objective. In order to optimize the objective function, we use the following two steps iteratively until convergence. The first step is to update the association matrix W while fixing S and G. And then we use W to help us do the refinement of S and G. And here we should keep the two inconsistency terms decrease so that the algorithm will converge. To achieve that, we can maintain, for example, we can maintain a fixed number of edges in S and G. For example, in order to update G, we can simply swap edge I prime, J prime, and the edge ij. And when the following inequality holds. And also for the second step, we can further take into a consideration of location and pathway information. The intuition behind it is that if two SNPs are locationally close to each other, they are more likely to have interactions. And similarly, if two genes belong to the same pathway, then they are more likely to have interactions. For evaluation, we first do the simulation study. Specifically, we generate 10 different gene expression profiles using the formula below. And the formula contains three parts. The first part is the signal part, which is the multiplication of the weighting matrix W and the SNP matrix X. And the second term is used to model the confounding factors. And the last term is for the noise. And the subfigure shown here stands for the ground truth input for the association matrix used. And the x-axis stands for the gene expression, and the y-axis stands for SNP. And here we have 100 SNPs. And obviously, we can see that there exist three groups of genes. But in order to simulate the incompleteness of the prior knowledge, we only feed the algorithms with the grouping information of the first two groups of genes, and leave the last group information of genes not be used for all the methods. And the results reported here. So we will see that the results reported by GGD-Lasso is more consistent with the ground truth, especially for those results for the last group of genes. We will see that the method with dual refinement outperforms alternative methods for a large margin. And we also quantitatively compare the performance of the method. So these figures visualize the ROC curve of the false positive rate and the true positive rate. And here strength stands for the values used in the weighting matrix W for generating the gene expression data. And we will see that for all cases, the proposed model outperforms those alternative competitors. We further study the robustness of the proposed model. And here we have two different kinds of noise. The first noise is used in generating the gene expression data. And we use SNR, which stands for the signal noise ratio, to denote how many noise exist. And the second kind of noise is in the prior networks. We will manually input some noise in the prior knowledge networks S0 and G0. And the X-axis for all plate stands for the ratio of noise exister in the prior networks. And we can clearly see that Dual Lasso, GGD-Lasso, is more robust than the G-Lasso. And with the increase of the ratio of signal noise, with the decrease of the signal noise ratio, we will see that G-Lasso is even more sensitive to the noise existing in the prior networks. So we can conclude that the dual refinement is very important for the robustness of the algorithm. We also evaluate the methods using the real data. Here we choose to use the East eQTL dataset. After the pre-processing, we obtain about 1,000 SNP markers and around 4,000 gene expression profiles. And we use the genetic interaction network and the PPI network as S and G, respectively. And we use cis and trans enrichment for evaluation. The cis enrichment analysis will contain two steps. The first step is to do a one-tail Mann-Whitney test on each SNP for the cis hypothesis. And the second step is to do a pairwise Wilson sign rank test on the p-values obtained by the first step for the two methods, and to see if the p-values for the two methods are significantly different or not. And trans enrichment is similar as that of the cis enrichment. The difference is that here we will use genes regulated by transcription factors as the transacting signals. And this table reports the results of cis and trans enrichment. And each value of the table is the p-value, and which is used to evaluate to what extent the method in the first column outperforms the method in the first row. The smaller the value is, the more significant that the first column method outperforms the first row method. And we see that the order of the table is consistent with that we have observed in the simulation study. Here, we will see that the GGD-Lasso outperforms GD-Lasso because GGD-Lasso further used some prior knowledge about location information and the pathway information. And GD-Lasso outperforms G-Lasso because the dual refinement will make the algorithm more robust to noise. And we also find that all the methods integrate prior network knowledge outperform that do not have. For example, LOIS and the Lasso method do not consider the prior knowledge. And also we can see that LOIS outperform Lasso because LOIS further considered the confounding factors. And we also report the gene ontology analysis results for the top 15 hotspots detected by GGD-Lasso. And we will see that all the 15 top-ranked hotspots detected by GGD-Lasso can be significantly enriched by some gene ontology category. Here, 1 star, here in the third column, 1 star means that the p-value is smaller than 10 to minus 3. And 2 star means that the p-value is smaller than 10 to minus 5. And the 3 star is more, even more smaller. So we will see that all those 15 hotspots detected can be significantly enriched. And we also report some the corresponding hits by other competitors. And we found the hotspot 12 cannot be detected by G-Lasso method. Here the hits is 0. And the hotspot 6 cannot be detected by SIOL. And the hotspot 3 cannot be detected by LOIS. We also summarized the number of significantly enriched hotspots here. And we found the GGD-Lasso reports more significantly enriched hotspots than other competitors. And also the ratio of significantly enriched hotspots of GGD-Lasso also outperformed the competitors. This will indicate that the GGD-Lasso will report more precise results than others. To conclude, in this paper, we proposed a novel and robust model to consider both prior knowledge of relationships between SNPs and relationships between genes. And also, the model considers the incompleteness and the noise existing in those prior knowledge. Further, we take into consideration of other information like the location information and pathway information.